I've been asked in the break that I'm not directly addressing in the course of the lecture. So one Shiv just asked, which is, what's the role of CD8 T cells in autoimmune disease? What's the example? So the best example for a role of CD8 cells in an autoimmune disease is, I think, in type 1 diabetes, where CD8 T cells directly kill islets, and there are transgenics and so on and so forth. And yes, of course, it's in the mouse, because the only way to prove that CD8 T cells are important in humans is to deplete the CD8 cells and show that the disease goes away, and you're never going to do that experiment. So a lot of this is very difficult to prove in humans. But in mice, the evidence is pretty definitive that CD8 T cells are involved in type 1 diabetes. Most autoimmune diseases in humans are caused by some combination of autoantibodies and inflammation, which is typically mediated by CD4 helper T cells, some combination of Th1s and Th17s. Both of those reactions, high affinity autoantibody production and cytokine-mediated inflammation, are dependent on CD4s. So the conclusion that the community has reached is that the central control point in autoimmunity lies in CD4 T cells. When they get messed up, there's trouble. Sometimes the trouble is autoantibody, sometimes it's inflammation, many times it's a combination of the two. But it's really failure of tolerance, we're going to define tolerance, in CD4 T cells that underlies the majority of autoimmune diseases. The experimental systems studying autoimmunity have focused on CD4s, and the therapeutic approaches for autoimmunity have emphasized CD4s. So that's sort of a big picture, that there is evidence in some diseases that other cell populations like CD8 killer T cells are involved, but really the focus is on CD4 help. That's where control fails. That's number one. Second point somebody asked me was, what's the contribution of memory cells to autoimmunity? Because obviously when your patient with rheumatoid arthritis comes to you, by now the reaction has been going on for a while, and you would expect that there are memory cells present. And amazingly, we don't know the answer. Because remember, there are two possibilities. One is that it's the preformed memory cells that are causing the damage, right? The other possibility is that in order to propagate a disease, you constantly need to recruit new naive T cells into the reaction. So the constant recruitment of naive T cells is required to propagate the disease. The reality, like in many, many situations, is that it's not either or. It's probably both. But it's very, very difficult to prove the relative contribution of naive T cell recruitment and the presence of memory cells. You can stain tissues or lesions and look for the existence of these cells, but the only way to prove that something is important is to get rid of it. And there are no convincing ways of selectively eliminating naive versus memory cells. So it's a really difficult question to answer. Usually, when you end up in a situation like that, they're probably both involved. But how are you going to prove it? Especially in a human disease. You don't have a prayer. And we still don't know the conclusion, the definitive answer. So that was the second point I was going to make that somebody asked me. The third was something that I avoided during the lecture on co-stimulation, which is, where does co-stimulation come from in an autoimmune disease? Because it's an immune response. In fact, it's a T cell response. Like all T cell responses, you should require co-stimulation. So where's the co-stimulation coming from? And we, labs like mine that are very interested in T cell mediated autoimmunity, think that there are two not mutually exclusive possibilities. One is that infection or tissue injury might lead to local co-stimulation. So if you have a viral infection in the pancreas, or if you have damage to the pancreas, that might provide local co-stimulation. The other, which I find particularly interesting, is that in order to get autoimmune disease, control mechanisms have to fail. If your control mechanisms are working, you're not going to get autoimmune disease, whatever those control mechanisms may be. So it is conceivable that when control mechanisms fail, you don't need a lot of co-stimulation. The response goes without much stimulus. And that's conceivable. It's also extremely difficult to prove one or the other. And it is especially difficult to prove any of this in human autoimmunity. So, just so you understand the theory, that co-stimulation may come from infection or tissue damage, and you may not need a lot in an autoimmune disease because control mechanisms have failed, so it's easier to activate. And it's all quantitative. And so that leads me to another general comment that, but even in a very short course like this, if you really step back and when you absorb some of this information, when I started in the morning, I said that you can probably map the steps in an immune response from the entry of the microbe to the elimination of the microbe pretty well. And that's what immunologists have successfully done. That we have understood the steps, a lot of it based on mouse, but wherever it's feasible to translate to humans, we've done that successfully. So both mouse and human data, wherever it's feasible, have allowed us to map a normal immune response. In striking and somewhat tragic contrast, if somebody asks you, why did my friend get MS or lupus or type 1 diabetes? You basically don't have a clue. You don't know the answer. And it illustrates the problems we all face in understanding complex human diseases. So we all feel that the underlying problem in understanding, the underlying problem in autoimmunity is a failure of control. So much of the emphasis in the last 20 years or so, 15 or 20 years, has been in understanding control mechanisms. And that's what I'm going to focus on. Even after we've done that, where we actually understand control mechanisms pretty well, if you ask the question, in such and such autoimmune disease, which of these mechanisms have failed, we have pretty lousy answers. It's really tough. So in the last the first 3 quarters of this lecture, I'm going to focus on why do you not respond to self? What are the control mechanisms? And in the last 15 minutes, I'm going to talk about the sorts of approaches that people are using to understand autoimmunity. Why do these mechanisms fail? Realizing that there's a lot we don't know. And when we'll get the answers, I don't know. So this is one of the big unknowns in the field. Yeah?
One question. We have been associating occurrence of autoimmune disease to HLA antigens.
I'm going to come to HLA and all of that later on. What was your question?
The question was the control mechanisms are failing. So is there any association you found with
I'm going to come to HLA later on. So we're going to start by talking just about basic mechanisms of regulation. And there's some really cool stuff that's happened in the field. I mean, it's so interesting, it's actually turning our understanding on its head. And then we're going to switch to autoimmunity and see what we can answer, but as I said, not much. So we're going to focus on the principles of immune regulation and self-tolerance, and I want to define self-tolerance in a minute. And when we talk about tolerance, there will be a big emphasis on regulatory T cells. And then at the end, we're going to talk a little bit about the pathogenesis of autoimmunity. So this is the fundamental problem that immunologists all try to deal with, that your immune system lives in a state of equilibrium. You're trying to activate it, and that's what we've been talking about so far, and you're trying to activate it to get rid of pathogens, but when that goes haywire, it's the same activation arm that gives you disease, inflammatory disease, and therefore you're constantly trying to control it. You don't want to overreact against pathogens, and you never want to react against self. And so this is the balance, and you live in this balance all the time. And when this balance fails, specifically when control mechanisms fail, that's when you get inflammatory disease. So that's the importance of regulatory mechanisms, to prevent inappropriate reactions against self, and to make sure you don't react too much to bugs. You react as much as you need to. That's much easier, because you react against a bug, you get rid of the bug, the stimulus is gone, and everything calms down. So that's easier to understand. This is really the big challenge. Why do you not react against self? And so that's, as I said, what we are going to focus on. The concept of not reacting is called tolerance. Tolerance, by definition, is unresponsiveness to an antigen that is induced by exposure to that antigen. It is not lack of response. Lack of response is passive. If you don't see an antigen, you don't respond. That's not tolerance. That's just nothing happened. Tolerance is an active process. You see an antigen in some way that shuts you off forever, that you will never respond to that antigen. And by definition, it's antigen specific. So it is different from immunosuppression. You give a patient cyclosporin, you shut everything down. That's immunosuppression. That, by definition, that is not tolerance. Tolerance is an antigen-specific phenomenon. You give an antigen in ways that shut the response down. So why is it important? And this is really the reason why it's important, that all of us, in fact, this is how the nomenclature arose. The idea is that you have learned to tolerate yourself. Hopefully also psychologically, I always point out. Not just immunologically, but most of us have learned to live with ourselves. So that is the idea of tolerance, that you've learned to tolerate yourself or live with your antigenic self. And that concept is called self-tolerance, and breakdown of self-tolerance leads to autoimmune disease. But once we understand how you can induce tolerance, the therapeutic potential is enormous. In theory, I can treat any autoimmune disease if I know the antigen. There are currently, in theory, I can treat allergies or any other abnormal immune reaction. And the allergists have been doing this for a long time without understanding any mechanisms. At least some of you in this room have gone through desensitization, or it's called specific immunotherapy or peptide immunotherapy. If you have a strong reaction against pollen, you get little doses of ragweed antigen in order to shut your immune response down. So that's one form of tolerance. There are now clinical trials being launched, multi-center trials in MS and type 1 diabetes, using antigens from myelin and antigens from islets and giving them in ways that we hope will induce tolerance. So this idea of inducing tolerance to treat immunological diseases is a big deal. And I referred to this in the morning, that the same idea is going to be relevant to gene therapy, systemic gene therapy, stem cells, et cetera, et cetera. So the therapeutic, although this is why we immunologists care. We care about tolerance because it's why you don't react against self. The therapeutic potential of tolerance goes much beyond that. And this is why it's getting a lot of interest over the last 10 or 15 years. Okay, so we're not going to talk about therapeutics and tolerance induction anymore. What I want to focus on is this first question. Why do you not respond to self? And so immunologists have conveniently divided mechanisms of self-tolerance into what they call central and peripheral. And central tolerance is what happens in the generative lymphoid organs, which are, the generative lymphoid organs are thymus for T cells and bone marrow for B cells. If you're actually paying attention, it says it on the slide, but that's okay. Most of you don't read the slide before I ask you the question. So it's the thymus for the T cells and the bone marrow for B cells. And I'll tell you what happens in those organs in a minute. The generative lymphoid organs are also called the central lymphoid organs. So this, what happens in those organs is called central tolerance. And then there are mechanisms that happen in peripheral tissues, and those are called peripheral tolerance. So what happens in the thymus and the bone marrow? And let's just think about the thymus for a minute. Although the picture shown here is of B cells because there's no dendritic cell presenting antigen, there's no MHC and so on. So the picture shown is of B cells, but that's a little bit of poetic license. Let's just think about T cells for a minute. So T cells come into your thymus, precursors, and generate millions of specificities. Does that process know what is self or foreign, yes or no? Does the process, remember, specificities are generated by recombination of antigen receptor genes. You take your antigen receptor genes that are like this, and you combine this with this, and this with this, and this with this, and so on. And that's all happening as a molecular process. Does that process know what's self or foreign? It cannot, because there's no receptor out there. You're generating the receptor, right? So the process of receptor generation has to be unbiased. Correct? It's not surprising that when you do that, you generate tons of lymphocytes that are specific for self. Now, in your thymus, let me do it the other way around. How do you get a foreign antigen into your body? 3 major routes. How do microbes come into your body? You eat them, touch them, and breathe them. When you do that, where do they go? Where do they go? Say it. Lymph nodes. To your lymphoid organs. Do they make it into the thymus? No. There is no bugs in your thymus. The bugs go to the lymph nodes in the spleen. So there are no microbes in the thymus. So what can you see in this thymus? Mostly self. Now I'm going to tell you an incredibly interesting mechanism by which self-antigens are displayed in the thymus. But mostly what you see is self. With me so far? The foreign stuff is going somewhere else. It's going to the lymph nodes. The same is true of the bone marrow. What you see in the bone marrow is mostly self. So when immature lymphocytes see self-antigens in the thymus and the bone marrow, yeah? Well, I'm going to come to that. How about self-antigens that are not expressed in the thymus? I'm going to come to that. So when immature lymphocytes see self-antigens in the thymus, whereas mature lymphocytes that see antigens can get activated, immature lymphocytes that have receptors for self, they see self in the thymus, they croak. They die. So this is what immunologists have called deletion or negative selection. The cells get selected out. Your collection of lymphocytes gets purged of self-reactivity. With me so far? That's one, and that's a major mechanism. The second, which is peculiar to B cells, is that if an immature B cell sees self-antigen, it will reactivate its receptor rearrangement machinery and generate a new receptor. It will change the self-reactive receptor and put a new receptor on the surface. This process is called receptor editing, although it was first described in highly experimental mouse models, it turns out, can anybody in this room guess how many of your circulating B cells have edited the receptors? Anybody know? 1%? 0.1%? More than half. More than half of your B cells started life as self-reactive. And in your bone marrow, they edited the receptors in order to survive. It's absolutely extraordinary that half of your B cells started off as being self-reactive. It just shows how lousy the process of receptor generation is, but it makes the point that if you're going to generate the maximum number, there's no quality control. You have to generate the maximum number and then select them out. There's nothing else you can do, is to have the maximum number and then select them. So half of your circulating B cells are estimated to have edited their receptors at some time in their early life. In T cells, there is, and that's never been shown in T lymphocytes for reasons that I'm not going to go into. In T cells, there's an alternative fate that I'm going to come to, which is some of the T cells don't die when they see self, they turn into regulatory cells. They turn into harmless cells that suppress immune responses. So that's 3 different fates: death, receptor editing in B cells, and generation of regulatory T cells, peculiar to T cells, of course. Now, as somebody said, not all antigens are present in the thymus. I mean, maybe that antigen in your left big toe is not present in the thymus. Well, you can't do central tolerance then. And that makes the point that this process is imperfect. It's pretty darn good, but it ain't perfect. That's why you and I and all of us are walking around with some frequency of self-reactive lymphocytes in our periphery. It's very difficult to estimate the frequency. There have been recent papers looking at humans from Mark Davis, and Mark is going to speak in a plenary session here, suggesting that the frequency of self-reactive T cells in humans, naive self-reactive T cells in healthy humans, is as high as the frequency of virus-specific T cells. There's a bucket load of trouble out there. And it's an extraordinary statement that although this process is fantastic, central tolerance, it ain't perfect. And all of us are walking time bombs because we have self-reactive lymphocytes floating around in our bodies. And it's the importance of the control mechanisms that stop them from getting activated. And so Mark, you should hear Mark's talk. It's coming up in a couple of days. I don't remember which day it is, maybe Friday, one of the plenaries. So that's central tolerance. And if that doesn't work because the T cells, the lymphocytes didn't see antigen, then they'll go off into the periphery and they get shut down. And that's peripheral tolerance, and I'm going to come to that. So I'm going to take a T cell-centric view. So this is what happens in the thymus. Here's a T cell that has not fully matured. In an immature state, it sees a self-antigen, and one of 2 things happen. A lot of them will die, and some of them will turn into regulatory cells that themselves cannot cause damage, but in fact will suppress responses to that antigen. So you've basically eliminated trouble. For anything you saw in the thymus, you've eliminated the potential of trouble. With me so far? We still don't know what determines this choice. Why do some die and why do some turn into regulatory cells? We don't know the answer. I'm not going to take any questions on regulatory T cells now because in about 10 minutes I'm going to come to those, 10, 15 minutes. So hold the questions about what regulatory T cells are. Yeah? So you're one step ahead of me. Again, you're segueing into my next slide. So the next slide says, well, what are the self-antigens you see in the thymus? And so some of this is no big deal. I mean, if you have a cell surface antigen that's present in all cells, it's also going to be present in the thymus, right? What we call ubiquitous, things that are everywhere, are going to be present in the thymus. No big deal. Easy to understand. It turns out that the thymus has an incredibly interesting mechanism for sticking self-antigens into the thymus. And that mechanism is really cool, in part because it's unprecedented, and in part because it was discovered by the study of a human disease. The human disease is called autoimmune polyendocrine syndrome, or APS1. A group in Finland found several families with this disease and did a rather heroic positional, because this is the responsible gene is autosomal. So both copies of the gene have to be messed up in order to get disease. And this is a heroic cloning effort, a gene identification effort. They did it with these families. They identified a gene, and they called it air regulator. And then Mark Anderson, who's also one of the focus leaders, who, when he was a postdoc at the Joslin, he knocked air out in mice. And when he did that, he absolutely reproduced the human disease. And they then went on to do a series of really elegant experiments. An old version of this slide is in your notes. We just made this new version of the slide for the next edition of our textbook, and it actually illustrates what air does much better. So it turns out that air is a transcriptional regulator that induces the expression of many tissue antigens. These tissue antigens are normally made in other tissues. For example, insulin is made in your pancreatic islet beta cells, right? Under the control of the glucose-sensitive insulin promoter, right? With me so far? But it's also made in the thymus under the control of air. And it's absolutely extraordinary that this one transcription factor makes the thymus an immunological window of yourself. It sticks a whole bunch of antigens into the thymus. Now, there are lots of molecular mechanisms we don't understand. But what if air is mutated? Now you don't express, and then once these self-antigens are expressed in the thymus, they are recognized by self-reactive T cells, and the self-reactive T cells are deleted. So that's the normal process. What if air is mutated or not functional? It turns out that these tissue-restricted antigens are not expressed in the thymus. If it's insulin, is insulin still expressed in your pancreas? Yes or no? Yes, because in the pancreas, it's not under the control of air. It's under the control of the insulin promoter. And so the tissue antigen is not expressed in the thymus. Self-reactive T cells are not deleted. They wander off to the pancreas, and they cause type 1 diabetes. And so in the absence of self-antigen expression in the thymus, self-tolerance fails. And that's the message. And air is one transcriptional regulator that controls the expression of self-antigens in the thymus. And it is an incredibly interesting story. By the way, I don't know if Andy mentioned this. These patients get candidiasis. This is another really cool story. Why would they get candidiasis? Why would you get candidiasis in an autoimmune disease? They don't get TB. They don't get viral infections. They just get candidiasis. So now Andy mentioned, and I don't know if you guys paid attention, but which cytokine or T cell subset do you really need to protect yourself against Candida? It's IL-17, right? So why are these autoimmune patients getting Candida infection? Because they make autoantibodies not just against their islets, but they make autoantibodies against IL-17. It's the first clearly documented, clinically significant example of autoantibodies against your own cytokines. And when the two papers came out saying, because all of us had been puzzled, we should have known, because we knew 5 years ago that you needed IL-17 to protect you from Candida. And this is an autoimmune disease with autoimmune attack against lots of self-antigens. And when the two papers, back-to-back papers came out that patients with this AIR mutation have autoantibodies against their own IL-17, all of us who read the paper said, duh. I mean, it was just such an obvious result that nobody ever really looked at and thought about. Now, of course, everybody is very much tuned into this link between autoimmunity and immune deficiency. Is it because people are making autoantibodies against useful molecules? So this has become a big deal, but this disease is really the poster child. Okay, so this is just stating in words what I already said, and I'm not going to go into it. So the general concept of peripheral tolerance is this, and again I'm taking a T-cell centric view, just in the interest of time. And this idea you've already heard, that if you get two signals, you get a good immune response, but if you recognize antigen and and don't get signal 2, instead of responding, you either fail to respond or you shut the response down. You become tolerant. The cell cannot respond again. So, that was the question, what is delivering signal 2 in the pancreas in the AIR mutant patients or in any other autoimmune disease? And we don't know the answer. It's either some level of cell death that's happening all the time, but the important point is, or as I said at the beginning, if you take away control mechanisms and you have a bucket load of self-reactive T cells, then maybe you don't need much second signals. Just a little bit will do the trick, and we don't know the answer. But if you remove self-tolerance mechanisms, then perhaps it's easy to activate cells because there's just a lot of them present. And that's a question that all of us struggle with. I mean, all of us work with mouse models of autoimmunity in which we don't immunize mice. We have a self-antigen and we do something and the mouse gets autoimmune disease. Where's signal 2? There's no adjuvant there. It's just normally present. And it's a really hard question to answer. So what are the mechanisms of peripheral tolerance? So immunologists have divided these mechanisms into 3 broad groups. T cell sees antigen, doesn't get all the right signals, it shuts itself down. It cannot respond again. It's not dead, but it's cell intrinsic unresponsiveness, right? And that's the phenomenon that immunologists have called anergy. The cell cannot respond again. Or the T cell sees self-antigen and regulatory T cells shut it down from the outside. So this is cell extrinsic, and that's what we call suppression. Or the T cell sees self-antigen in the periphery and it croaks, it dies. And that's what we call deletion. So the 3 mechanisms of peripheral tolerance are anergy, suppression, and deletion. Yeah? It's a complicated question, is anergy reversible or not, and I'm going to come to that very briefly. I don't want to spend a lot of time on anergy. So what are the mechanisms of anergy? Why does a cell say, I'm not going to respond again? And people postulate that there are 2. One is a signaling block. So in an allergic cell, the T cell receptor just doesn't work. It stopped working. Why would that happen? Well, there are many possibilities, because one possibility that has been raised is that when your T cells see self-antigen, they activate these enzymes called ubiquitin ligases. And these ubiquitin ligases attach ubiquitins to many of your signaling molecules, perhaps CD3, perhaps zeta chain. And if you attach ubiquitin to a protein, what are you doing to that protein? What are you instructing that protein to do? I'm sorry, I didn't hear that. You're targeting that protein for destruction, for enzymatic destruction. So maybe self-antigens for some reason turn on ubiquitin ligases that chew up signaling molecules and just stop the cell from working. The other mechanism of anergy is this one, and I referred to this earlier, that some self-antigens might induce CTLA4 or PD-1 or one of these inhibitory receptors, and once that happens, it shuts the cell down. It doesn't kill it, but it shuts the cell down. So, it's a poorly defined phenomenon, and there are lots of questions that are unanswered. To me, a lot of this has been done in mouse models. To me, the biggest unanswered question, and that's what this says, that a lot of it has been done in mouse models. The biggest unanswered question is this second one. Are you and I walking around with energetic cells specific for self-antigens? And how do you prove that? Unless you have assays for antigen-specific lymphocytes, how do you prove whether you or I? And so that's the question. Do natural self-antigens in you and me induce anergy? And actually the answer is we don't have a clue, because there are no good markers for energetic cells. We don't do the right assays. So nobody can answer that question. So the phenomenon is extremely well described in animal models, but its relevance to humans is extremely poorly established, because the correct assays are extremely difficult to do. I'm going to, because we're running short of time, this lecture will go a little bit long because I want to sort of touch upon autoimmunity also. But we're running a little bit, well, no, I'll tell you the next story. So I'm not going to go into pathways of cell death. I don't know if this is in your notes or not. I may have added it later on. It is in the notes. So there are multiple pathways of cell death. One of those pathways is the expression of a death receptor called FAS, which is shown here, and the ligand for FAS. And when the FAS is a member of the TNF receptor family, and when that happens, the cell kills itself. And its importance was also demonstrated by the study, just like the importance of negative selection in the thymus was demonstrated by the AIR mutants. The importance of this death pathway was also demonstrated by a collection of mutants in mice and in humans. And this is the story of a little girl, and these patients were seen, they're usually between 1 and 2 years old. They were seen almost simultaneously. They were not just seen, but the mechanism was understood in Paris and at the NIH. These are little children who come in with big lymph nodes and spleen, so they're referred to their pediatric oncologist because you think that the pediatrician thinks that they have a leukemia or a lymphoma of some sort. And then you work them up, and they have lots of evidence of autoimmunity, including anti-nuclear antibodies, many signs and symptoms of autoimmune disease. And so this is a peculiar phenomenon, and then the physicians who were looking after them were also immunologists, and they realized that exactly the same phenotype had been described in mouse models. And in mice, the relevant gene had been identified. The relevant gene is this death receptor called FAS. And so they went looking for FAS mutations and found them. So this is a disease, an autoimmune disease caused by mutations in FAS that lead to a failure of apoptosis or cell death in the periphery. It was the first disease known in which defective cell death led to an autoimmune phenotype. There's not much you can do because you can't put normal fast back into these kids and cure them. So basically, they're treated by bone marrow transplantation. It's been a powerful result, but these are rare diseases. They make the point that peripheral deletion must be important because if you mess it up, you get autoimmune disease in rare situations. Yeah. That's a very interesting, and that is the phenotype that was peculiar. A lot of the T cells in these patients have neither CD4 nor CD8. And the mice are exactly the same. That's what people saw when they said this is similar to the mouse models, and nobody has a clue. We've still not. We spent 10 years working on FAS in the old days, never figured that out. Had all kinds of interesting hypotheses that went nowhere. Okay, so the last 15 minutes I want to talk about regulatory T cells. Regulatory T cells, we think most of them arise in the thymus by recognition of self-antigens, but we think that they can also arise in the periphery by recognition of self or perhaps foreign antigens, like tumor antigens. And I'm going to define the phenotype in just a minute. Regulatory T cells work mainly by shutting down T cell responses, but they can also shut down other responses. And I'm going to come to how they shut down these responses in a minute. So what are these cells? The best defined regulatory T cells are CD4 cells that express high levels of the IL-2 receptor and, most importantly, stably express the transcription factor called FOXP3. And the reason why I say they're the best defined is that mutations in FOXP3 lead to a failure of self-tolerance. Mutations in FOXP3 lead to defective regulatory T cells, a failure of self-tolerance, and lethal systemic autoimmune disease. That is definitive proof that FOXP3 positive regulatory T cells are important. If you can demonstrate something in a genetic model that is definitive proof that it must be important. With me so far? If you read the literature, there will be hundreds of regulatory populations defined. For none of them have people proved that if you get rid of them, you get autoimmune disease. So this is the one for which the proof is the best. That doesn't mean they're the only cells that suppress, but it says they are the most convincing regulators of immune responses. That's true in this disease. The disease is called IPEX. Some of you know about it. It's immune dysregulation, polyendocrinopathy, again, endocrine organs. Enteropathy, these kids develop colitis, and it's X-linked. So that's what the acronym stands for. This is a rare disease. The big question out there is, in common autoimmune diseases, like lupus and MS and rheumatoid arthritis and what have you, is there a defect? Is there a defect in regulatory T cells? And amazingly, we don't have a convincing answer. It's been really tough. If you go back and look at all the published papers, some say the numbers are down, some say the numbers are normal, some say they don't work very well, others say they work fine, some say that the effector cells are resistant to suppression, and others say the effector cells are fine. It's all over the place. So, although we say that many autoimmune diseases may be associated with defects in or resistance to regulatory T cell mediated suppression, the truth is that in human autoimmune diseases, we don't have a clue. And it has been a nightmare of a problem to answer. So all of you who work on autoimmunity will have your own data. All of you will have strong opinions. And there's somebody out there who'll disagree with you. And you take it for what it is. I mean, I don't have an answer. So if you ask me the question, are common autoimmune diseases associated with defective Tregs, I don't have an answer. And that's basically all you can do at this point. That doesn't mean you should stop studying it. It just means we ain't got an answer. I'll tell you towards the end why I think we don't have an answer, but we'll come to that in a minute. There are many mechanisms of action that have been proposed. The most recent review article on regulatory T cells had 22 possible mechanisms of action. I think only 3 are important, 2 of which are stated here. The third should be. One is CTLA-4-mediated suppression. So CTLA-4 on the Tregs just eats up the B7 from dendritic cells, and we talked about that. One is the production of cytokines that shut down immune responses, like IL-10 and TGF-beta. And the third is that regulatory T cells express very high levels of CD25, which is the IL-2 receptor, and so they just eat up, they sop up IL-2. They deprive the environment of IL-2. And all 3 have been demonstrated in vitro and in vivo. So there's evidence for all of those mechanisms. So you should add consumption of IL-2 as the third mechanism. One very interesting result. If you knock out CTLA-4 in FOXP3 positive cells, follow me, only in regulatory T cells, the mouse gets systemic autoimmune disease. If you knock out IL-10 in FOXP3 positive cells, the mouse gets only colitis. So that says that some of these mechanisms are more important in the gut and others are more important everywhere else. But that's basically all you can say. That result is definitive. It's a knockout mouse result. That if you knock out CTLA-4 from these cells only, you get systemic autoimmunity, you knock out IL-10, you get colitis, and nothing else. No other disease. So there are multiple mechanisms. Some may be working in one tissue, others in another. Are there subpopulations of regulatory cells? How heterogeneous are they? These are all open questions. One last point I want to make is that those of you who study humans will know that FOXP3 can be transiently expressed by many activated T cells, not necessarily regulatory T cells. So in the community, there is a big push to not just stain for FOXP3 expression, but look at demethylation of the FOXP3 locus. Because demethylation is an epigenetic mark for stable gene expression. So FOXP3 demethylation is becoming an increasingly accepted tool for studying this cell population. And it's really just a marker of stability. There are nomenclature problems in the literature, and Jeff Bluestone and I are partly responsible. So the ones that are generated in the thymus we call natural because they're present all the time, and the ones that are generated in the periphery we call adaptive because they're induced in response to some antigen in the periphery. And we now are also part of a nomenclature committee that met about 6-8 months ago that has strongly suggested, and it's like we're changing our own minds, has strongly suggested that you get rid of these terms, natural and adaptive, and call them thymic if you know they came from the thymus, and peripheral if you know they came from the periphery. Of course, the problem is that in you and me, I have no idea which Treg came from the thymus or came from the periphery, because there are no convincing markers. So what are you going to call them? So our strong suggestion is to get out of this nomenclature and just call them FOXP3 positive regulatory T cells and get away from all this thymic, peripheral, etc. In experimental models, you can prove that they came from the thymus and the periphery. In humans, it's really hard to do. And so just get away from this nomenclature mess. The markers, there are lots of markers that have been described, and none of them has withstood the test of time. And I'm not going to go into it in any more detail. Yeah? So is, you know, I think I've paid you guys. 2 slides and I'm going to come to IL-2. Everybody is sort of helping me segue into the next part of my talk. And so I want to actually present a concept, but I'm going to do it very quickly. And the concept is that if you induce or activate regulatory T cells, and this is something I'm going to talk about at at a lecture later on, so I'm not going to get into the science, but there's only one point I want to make. So the concept is that if you activate or generate regulatory T cells, some of them survive as a memory population. And these memory Tregs continue to suppress immune responses. And so we've proposed this as a concept of regulatory memory, and you don't have to go through all of this, it's in your notes. But I wanted to make one point. It turns out that one of the most common graduate student questions in immunology is, why doesn't the pregnant mother reject the fetus? Because half the fetus is foreign, right? And you've never seen that half. So why is the fetal graft accepted? And there are 2 papers that came out in 2012 that are really cool. So the first one said that if you follow through evolution, placentation, the ability of a species to develop a placenta, came at exactly the same time as the ability to generate stable Tregs, stable regulatory T cells. It's an absolute concordance. And it's a pretty powerful evolutionary argument. So if you look at pre-placental marsupials, like opossums and wallabies, you know, they don't have a placenta. Of course, I said this somewhere, and I've never looked up Wikipedia, and somebody said, well, if they don't have a placenta, how do they look after their babies? And I actually don't know the answer. I mean, you can see the pictures of those little ones hanging in those pouches. So these are pre-placental. They don't have a placenta. They can't generate stable Tregs. And if you look at the next step in evolution, they're capable of generating stable Tregs. So that alone is a pretty powerful argument that regulatory T cells arose in order to allow placentation to happen. And then there was a really gorgeous experiment done saying that in a mouse, that when a mouse sees a paternal antigen, the pregnant female then will generate regulatory T cells specific for the paternal antigen. Some of them will survive as memory cells, as memory Tregs, and they'll continue to protect against subsequent pregnancies. And if you get rid of them, then you get fetal resorption. So these were 2, they're not from us, they're not from my lab, they're 2 really gorgeous papers that suggest that the ability to generate long-lived regulatory T cells is what leads to the protection of the fetal graft. It leads to lots of questions. Are there defects in this phenomenon that are the basis for recurrent spontaneous abortion, so on and so forth? And we don't know the answer. But I'm going to leave you with the idea that maybe we are finally getting some answers to why the pregnancy, why the fetus is not rejected. So there's been an explosion of information about Tregs, generation, properties, etc. And the big question is, can we possibly do cellular therapy? And so there are many ongoing attempts. There are actually hundreds of clinical trials, either in the works or being planned, taking patients' regulatory T cells, expanding them ex vivo, and putting them back into patients. And actually, a few of these now are beginning to get reported. So at first it was a real fear. You know, the fear was, so how do you know these regulatory T cells won't turn into Th17s if you put them back into a patient with autoimmune disease? You actually didn't know that. And if you actually think about it, this was an incredibly gutsy clinical trial, because some of these were done with kids with type 1 diabetes. And you know, you can treat kids with type 1 diabetes with insulin. So now you're taking their own Tregs, sticking them back into these children. They're usually somewhere in the sort of the 10 to 15 age group, so they're children. You're putting their own Tregs back, and you don't know what the fate of those regulatory T cells is going to be. If they all turn into Th1s, you'll absolutely make the disease worse. So the good news is that that didn't happen. They put large numbers of Tregs in culture expanded Tregs back. It's safe. The benefit is very transient. These regulatory T cells hang around for a while. They seem to provide some benefit, but then they probably just disappear. So for 3 or 4 months, the insulin requirements come down, and then they go back up again. And so this is what we are beginning to see in the very few clinical trials that have been reported. There are still many challenges, and I'm not going to go into Treg therapy. But I do want to come to interleukin-2. So this is what we know of interleukin-2, and I'm also going to talk about this with some real experimental data when I give my keynote address in a couple of days. So we don't need to spend a whole lot of time. So this is the experimental, this is the concept, the dogma about interleukin-2. It's made by CD4 helper T cells, and it stimulates the growth of CD4 helper T cells. So that's why it was called T cell growth factor. You need it in order to get an immune response, right? And so if you knock out interleukin-2 in a mouse, so it's a T cell growth factor, you need it to get an immune response. And so if you knock it out, the mouse won't get an immune response, and the mouse will be immunodeficient. Correct? Yes? Yes? God, you guys are really gullible. So I suckered you into that one because I joke with people that if all our predictions were correct, immunology would be a very boring discipline, and it's definitely not boring, because in fact what happened when you knock out interleukin-2 is that the mouse gets huge lymph nodes in spleen and drops dead of autoimmune disease. The exact opposite. In the absence of T cell growth factor, there are too many growing T cells. With me? With me so far? Without T cell growth factor, there are too many T cells. So this is a problem, right? It took 10 years, many false leads, including in my lab, wrong hypotheses that we wasted many person hours in chasing before the answer really came out that interleukin-2 does two things. It does stimulate an immune response, but it also maintains regulatory T cells. It's responsible for their survival and their function. What's amazing from the mouse knockout results is that its ability to stimulate an immune response must be redundant, because the IL-2 knockout mouse is making a whacking big immune response against self. Correct? So its ability to activate T cells can be replaced by something else. It's redundant. Second time I've done that. But its ability to maintain Tregs is not redundant. It's obligatory. And so that's what interleukin-2 really does. It maintains regulation. This is a redundant function. Hang on. Let me take this through one more reasoning. And so interleukin-2 was first used to boost immune responses in cancer. There was a very large and expensive clinical trial of interleukin-2 in HIV seropositive individuals trying to boost immune responses in HIV infection. Neither of those attempts, after probably a couple of $1 billion, at least a few $100 million, neither of those attempts has been a glorious success. And so people went back and looked at those patients and said, what's happened to the FOXP3 positive cells in these patients? And the answer is that the numbers go up. And so that led to two different groups about a year and a half ago, and now many groups and many pharma companies saying, can we give interleukin-2 in ways that will suppress immune responses? This was to boost, and now we're giving it to suppress. and successful proof of concept studies were reported at the end of 2011, I believe. So this is an incredible example of a field being turned on its head. The cytokine that you were using to boost immune responses, the same cytokine is now being used to shut down immune responses. It obviously has a problem. One sec, there is another question. It obviously has a problem. We know it does both. I just showed you that slide, that it does both. So now you've got to figure out, how am I going to give it to my patients that it does what I want it to do? And that's the big challenge in the field. Anytime you have a situation where an agent has two diametrically opposite effects, it's a tough problem to figure out how to control its effect. And that's where the IL-2 world is. But those of you in pharma and immunology companies should be aware of this, because there's a lot of interest in using IL-2 to dampen the immune response. And you can see the logic behind it. There was a question back there. The question is, do differentiated Tregs divide in response to IL-2? There's a myth out there that regulatory T cells are energic and don't divide. That is generally true in vitro, but there are numerous in vivo situations, particularly in mouse models, where they actually divide even more than effector cells. So that myth is an in vitro phenomenon and an artifact. We actually were among the first to show that Tregs proliferate just fine. And we've got recent data that under some situations they proliferate even more than FOXP3 negative cells. So this business of Tregs being energic is very much a culture dish phenomenon.
So why then there's this discordance between in vivo and vitro response?
Oh, there's not a discordance in vivo. Oh, so it's much easier to demonstrate the effects of interleukin-2 on regulatory T cells in vivo. Much easier than it is in vitro. A lot of what's been done has been IL-2 treatment. So all of us can do that. All of us now have protocols for giving IL-2 to a mouse that will increase the number of Tregs, and pretty selectively increase the number of Tregs. How well can we translate that to humans is the classic problem. And that's all I'm going to say, is that there are numerous efforts going on at the moment to translate this. But the concept you guys must understand, interleukin-2 is a necessary growth factor for regulatory T cells, and that's really what it does. You had a question. Correct. Correct. So, absolutely correct. That her comment is that these were done with very high doses, and now we are doing it in much lower doses. The idea is that regulatory T cells have a lot of the high affinity receptor, so they should respond to low doses. And these two trials were both, as you see here, done with low dose IL-2. But you know, it's tricky business. I mean, you're going to give it to patients and just hope that the dose is going to make a difference. This is tricky business. I mean, nobody wants to do that. If you were giving, just changing the dose of TNF to either cause good results or bad results, you'd be terrified of giving people TNFs, just based on the dose. So it's a tricky business to be relying on dose, and there are all kinds of stuff going on. So people, for example, are mutating IL-2 to ask, can I get a mutant that will do one versus the other? And there's all kinds of really cool science going on. What's really interesting is that, this is one of the first cytokines known, and the first one in the adaptive immune system. And it's as if based on mouse knockout data, so all of you who say the mouse isn't useful, based on mouse knockout data, we are completely revising what we think of IL-2. It's extraordinary, and it came straight out of the knockout mouse. There was not a single human experiment. Everything was done in humans after the fact. So it just shows you the power of experimental systems. It also shows you the challenge of going from the mouse to the human when it comes to therapeutics. But this is one of the really powerful results of how a field has been totally I want to skip that. I want to skip that. That's more philosophy. This is more philosophy than anything else. I do want to make the point, though, that I've talked to you about all these mechanisms of tolerance, air, FAS, FOXP3, etc. And I've told you that their importance has been demonstrated in rare genetic models. Now remember, genetic models are very powerful, because if a mutation in a gene leads to a phenotype, you know that gene is involved. I mean, you cannot question that. That's an absolute and definitive statement. But we still don't know. Do Tregs fail? Does energy fail? Does negative selection in the thymus fail in common autoimmune diseases? We don't know the answer. I can tell you for sure that Tregs fail in patients with IPEX. That's a few hundred patients. But I cannot tell you for sure that they fail in patients with rheumatoid arthritis or MS. So how to take these mechanisms of tolerance to autoimmunity remains an enormous problem. Despite that, what do we think of? So the definition of autoimmunity is pretty obvious: immune responses against self. It's worth remembering that much of our knowledge of all immunological disorders, and specifically autoimmune disorders, is based on mouse models. We are all concerned about the convertibility of mouse to human, but that's just reality. And there are many problems. Specifically, we don't know which mechanisms of tolerance fail in which disease. And this is, to me, a huge problem. So what do we think of when we think broadly about the mechanisms of autoimmune disease? So like this, what I'm about to say sounds very sophisticated, but as you'll see in a minute, it's a pretty trivial statement, because the conclusion that people have reached is that autoimmunity is because of some combination of susceptibility genes and some environmental triggers. And the two come together to give you autoimmunity. That sounds sophisticated, susceptibility, genes, and environmental triggers, but you realize that every complex disease, everything other than a Mendelian disease, is exactly the same. Cancer is susceptibility, genes, and environmental triggers. Atherosclerosis is the same. Allergy is the same. So everything is the same. It's a kind of a meaningless statement unless you can figure out what are these genes and what are these triggers. So that's where the world has been at, trying to identify the genes and trying to at least begin to ask what the triggers are. So we know that autoimmunity has a strong genetic predisposition. The most important message when you think about genetics of autoimmunity is that it's a multigenic disorder. The single gene defects that I've been talking to you reveal pathways. They're very instructive, but they do not tell you about common autoimmunity, because common autoimmune diseases are multigenic. So what are the genes involved? By far the most important, known for 50+ years, is HLA. We've known about HLA and its linkage. So remember, HLA is highly polymorphic. Polymorphic means that there are many variants in the normal population. Each variant is called an allele, so there are many alleles in the population, as many as 5,000 for some HLA genes. It's quite amazing, and we can talk about the biological implications of that. We have known that some HLA alleles are associated with some autoimmune diseases. HLA-B27 with spondyloarthropathies has been known for 50 years. Despite that, Most people with HLA-B27 will never get cervical spondyloarthropathy, so it's present in healthy individuals, and we have no clue. It's really kind of a depressing statement. We have no clue what B27 does in spondyloarthropathy. We have no idea whether it's presenting an antigen. I mean, it must be presenting an antigen. That's the only thing that HLA does. It's a peptide display molecule. What antigen? To what cells? And how are those cells getting activated? We have absolutely no idea. And HLA is strongly associated with autoimmunity. There are many, many non-MHC genes, non-HLA genes that have been linked to autoimmunity. The linkage or the odds ratios are much smaller. Some of them are potentially interesting, and they're listed here, and I'm not going to go into them. But it hasn't led, defining these or identifying these genes has not led to any real insights. And so this is a wide open field. The genomics, remember in the old days, the way this was done was what's called genome-wide association studies. And that's basically looking at nucleotide polymorphisms in 5,000 healthy people and in 3,000 people with a particular disease, and then comparing the frequency of polymorphisms in different regions of the genome. That's basically what was done. And many different polymorphisms have been identified that show this kind of association with autoimmune disease. But what they really have to do with the autoimmune disease, we still don't have an answer. So it's been interesting, but it's also, in terms of illumination, has been frustrating, because we still cannot link these genotypes to pathogenesis. There are also limitations of genome-wide association studies. By definition, they detect common variants. They detect the statistical frequency of common variants. But if you were to argue, or if you were to postulate, that maybe autoimmunity is a mutation, just like cancer. So if you think about it, if you were to do genome-wide association studies in patients with cancer, you'd never find RAS or P53 because you weren't looking at the tumor. But if you were to argue that maybe autoimmunity is a mutation in autoreactive T cells, maybe patients with multiple sclerosis have a mutation that hits myelin-reactive T cells, and only those, just like cancer, well, you'd never find it, because rare mutations cannot be detected by this kind of technology. So there are lots of limitations. I think the predictive value of these associations, they're so poor, the associations, that can you look at a bunch of people before they get disease and predict who's going to get the disease? The answer is no, because the associations are not strong enough. I mean, B27 has an odds ratio of 100, and even there you can't predict, of all the people who have B27, which ones are going to get cervical spondyloarthropathy. You have no idea, and there's no way to tell. So the predictive value of the genetics has been weak, and many, many companies got all hot to trot on the idea that if we can identify these associated genes, we can use them to develop therapeutic targets. The reality is that each gene makes such a small contribution that most companies are backing off from this, that they've realized that going from genomics of complex disease, this is not a Mendelian disease, this is not a single mutation, but going from the genomics of complex disease to actual drug development is going to be really tough, and has actually not succeeded so far. So more is going to be done. Now people want to do whole genome sequencing in thousands of patients with autoimmunity. We've probably spent somewhere between 500 million and $1 billion to do all these genome-wide association studies. If we do whole genome sequencing, now we'll increase that tenfold. But you know, hey, I mean, we sequenced the human genome because we could figure out the cause of human disease. That was one of the reasons. So it's a logical next step. Personally, I think that we understand these mechanisms by much more methodical hypothesis-driven science. These kinds of large-scale, high-throughput methods tend to be rather frustrating. But people tell me I'm old-fashioned. So I think you just have to take it, you have to make these decisions yourself. What's the right approach for understanding the pathogenesis of human autoimmunity? Very quickly, infections can trigger autoimmune reactions, and this is an example of infections leading to upregulation of co-stimulators. There's a lot of interest in the role of the microbiome. For those of you who don't understand the microbiome, remember what it means is that there are a lot of organisms that coexist with you, commensal organisms. They cannot be cultured. 95% of them cannot be cultured, so their identification required sequencing technologies. And when we developed those sequencing technologies, we discovered that there's a whole world of bugs out there that you didn't know about. Now, are they associated with autoimmunity? Again, in mouse models, maybe. In humans, the microbiome in you and me is incredibly variable. It changes from day to day. It changes from city to city. So will we ever be able to find out? Who knows? There's a lot of interest in doing this kind of study, but will this lead to answers? Wide open question at the moment. It's also worth remembering that some autoimmune diseases, although the focus has been on triggering, it's now pretty clear that infections protect people from some autoimmune diseases. And I'm not going to get into the hygiene hypothesis for the sake of time. It's really a hypothesis that's relevant to allergies much more than to autoimmunity. I can talk about it afterwards for you guys if you want. Many other environmental influences, another huge black box. As all of you know, many systemic autoimmune diseases are tenfold more frequent in women, and Sjogren's is even more than tenfold more frequent in women than in men. Numerous studies funded by the NIH and other organizations to try and understand the gender bias of autoimmunity. And at the end of the day, there are no answers. We don't know why. Lots of hypotheses, but very little understanding. So these are tough problems. And I want to make one point in a couple of slides about why I think we are not doing all the sorts of things we should be doing. Rather than go away with it's all bad news, I think that you should appreciate by now, and we're not going to go over all of these, but this is a quick summary on this one slide of all the therapies that have been developed in the last decade or so based on our understanding of fundamental science. So this is why you do research. If you understand these pathways, then you can actually do something about targeting these pathways. So these are all targeting at the late stage, at the stage of inflammation and tissue injury. We are not really understanding why the disease started, which is what we've been talking about. How do you understand why the disease started? By the way, I don't know if Andy mentioned this, but even here the clinical trials are revealing some surprises. So if you look at the clinical trials, you'll see that IL-17 and the Th17 pathway has been implicated in psoriasis and in IBD, specifically Crohn's disease, and in rheumatoid arthritis. The clinical trial data are incredible. It wipes out psoriasis. It works like gangbusters. A single dose treatment, and some of these patients are cured or in remission for 6 months. It has really questionable effects in rheumatoid arthritis. 2 of the 5 companies that have developed IL-17 antagonists say it works. 2 of the others say it absolutely doesn't work, so it's open. And Crohn's disease, in which IL-17 producing cells have been identified, mouse models have been done. Do you know what happens to people with Crohn's disease treated with IL-17 antagonists? Anybody? The disease gets worse. And what do? I mean, the mouse model said it should work. There's IL-17 in the gut. everything made sense, the disease gets worse. So sometimes it's actually the clinical trial that makes you wake up and reassess. Clearly, we've been misled about the role of Th17 cells in IBD. I don't know what's happening. I don't know what cells are involved, but clearly it ain't what we all thought it was. And these are really instructive, because another point I want to make to you guys is that really realistically the clinical trial is the only experiment you can do in a person. I mean, this is the only thing you can do to manipulate a human immune system. So sometimes they work exactly the way you'd have predicted, like IL-17 in psoriasis. And sometimes they throw you for a loop. And this is going to keep happening as we develop more and more of these drugs. So where are we with, yeah? It's not working as you think it. Who knows? Who knows? How many doses are you going to try? I'm sure all these trials have been done with dose escalation, so it's not a single dose. Who knows why it's not working? Not working. If you ask me, why is it not working? The answer is yes. How are you going to answer that question? So I can't answer that question. So where are we? And there's a point I want to make. So I think we've learned an incredible amount. We've learned an incredible amount about basic mechanisms of tolerance. We've learned a lot about disease pathogenesis. We don't know all the answers. We don't know what tolerance mechanisms fail in which autoimmune disease, but we've learned an incredible amount, and all of it we've learned, or much of it we've learned from animal models. I think what we as a community of immunologists have failed to do is develop strong technologies for studying people. For example, if you take your patient with rheumatoid arthritis, why do you want to study all the T cells? The only T cells you want to study are the ones that are attacking the joint. But we haven't developed the tools for doing that. Now, there is a huge push to do this, to learn how to study antigen-specific immune responses in healthy individuals and then take that to human disease. It's going to be expensive. The technology is tough. You have small cell samples that you're going to deal with. But at the end of the day, it's the technology that's going to give you the answers. And I think that's where the world is going. Throughout the meeting, you'll hear examples of modern technologies being applied to the study of human disease. And my sense is, this could be a cop-out, but I tell people as I get increasingly senior that my generation laid the foundations, which is the first bullet out here, and your generation is going to have to do the tough stuff. I think the foundations were a whole lot easier than studying human disease, but I really mean that in a serious way, that the next 20 years we now understand the basics and we're going to build on that and develop technologies for studying disease. So that's where the world is going. Yeah, it's a good point that there are very interesting humanized mouse models that are being developed in which the mouse's immune system is being gradually replaced with a human immune system. I think it's early days. You should pay attention to it, but how valuable it's going to be, we don't know the answer. At the end of the day, some people will tell you, you want to study a human? Go study a human. And there is some truth to that. You know, of course, the flip side is it's really tough. I mean, it's not going to be easy. But there is some truth. You want to study people, go study people. Don't futz around. And I think that the world is going to change. I mean, I'm really an optimist. You know, I can think back from the days that I was a postdoc, and I can now see how much we've learned about the immune system. How many of the principles are now firmly established. It's pretty darn amazing. It's mind-boggling what we've learned. So the next step is going to be the tough stuff, but the tough stuff is coming. Thank you all. Questions? Anybody else? Yeah. Yeah.
Where did we get the notes you've been talking about? Was it one of the zillion emails we got from Focus?
Oh, yeah. I think Lori sent an email link to the PDFs or something like that, like last week or something like last week. Yeah. Yeah. Yeah.
Is there an error-like mechanism for bone marrow?
Good question. Is there a mechanism that sticks antigens into the bone marrow? We don't know the answer. We don't know the answer. You know, you're going to have to luck out and find the gene, right? Yeah.
What do you think about the role of stress in the
You really had to ask that, didn't you? Personally, I think that psychological factors influence the symptoms, but my own bias is that there is no convincing data that they actually influence the immune response itself, other than high levels of cortisol production as a consequence of stress. But other things, do I really think that stress is important in the development of autoimmunity? No. It's your regulatory mechanisms are failing. That's why patients get autoimmune disease. It's got very little to... Now, stress may make their symptoms worse, but that's different from causation, right? So I don't think it's that important.
Does the estrogen and the progesterone play any role in the fetus antigen-specific production?
It's a good question. Do estrogens and progesterone have any role in the generation of antigen-specific Tregs in response to fetal antigens? And we don't really know the answer. You know, there's this very interesting experiment. You take a mouse that is carrying a fetus, a strain A mouse that's carrying a fetus from a strain B father, okay? So there's a foreign fetus in there, and that fetus will be accepted. You take the same strain B mouse, get a skin graft onto the pregnant female, and the skin graft will be rejected. In other words, acceptance is peculiar to the placenta. It doesn't happen in the periphery. So what's peculiar about the placenta? Nobody knows. It's a really interesting experiment. This is a 50-year-old experiment, that the mouse that is accepting the fetus from one strain of father will reject skin from the same strain. It's a very old experiment. It's a very cool result. If those Tregs are working, they should be working against everything. Somehow they seem to be working very well in the placenta and not elsewhere. Don't know anymore. Yeah?
Does the thymus do the same thing in adults as they do?
Yeah, that's another very interesting question. Does the thymus do the same thing in adults as... And so, you know, all of us don't have much of a thymus left because you start involuting your thymus after puberty. By now it's mostly gone. So what's happening with... Why aren't you all getting autoimmune disease if your thymus is gone? And you're obviously not. You know, most people don't get autoimmune disease. And so the likely answer is that there's enough of the thymus still there. You just can't find it in the fat, but there's enough of it there. There may be other answers. Some people have postulated that there are extra thymic sites where this happens. It's been very difficult to prove. So my own bias is that throughout life there's enough of a thymus. You can actually do an experiment in people. You can take a 50-year-old woman with metastatic breast cancer, and this used to be done, and then you ablate that person's bone marrow. You know, you harvest that person's bone marrow, cryopreserve it, and then give that woman very high doses of radiation and chemotherapy in order to kill the cancer cells, and then put the bone marrow back into that person, right? Autologous bone marrow transplants for metastatic cancer, this used to be done. It doesn't have much survival benefit, so people don't do it anymore. But this was done for quite a lot of people. And those people don't get autoimmune disease. They regenerate their full immune system. They don't get autoimmunity. So whatever thymus is there must be doing its job. And that's actually a human experiment that's been done repeatedly. So, you know, this business of the thymus disappearing, well, sure, it's hard to find because there's not much there. It's probably enough. There's a question in the back, and then we need to stop. I'm sorry? So it's an interesting idea. Do cyclosporin-treated patients have reduced Tregs and they tend to get relapses down the road? Maybe. But very, very difficult to do definitive studies in large enough numbers. There was actually also, there were these mouse experiments done some time back that mice treated with cyclosporin develop autoimmune disease. Yeah, when you stop the cyclosporin. And this is this very peculiar phenomenon that nobody quite understood. But that could also be a defect in IL-2 and IL-2 dependent Tregs. Nobody has revisited that. Okay, guys, enjoy the meeting. Yeah.
Question. Does the dendritic cells of the skin are different from the dendritic cells of the gut?
There are lots and lots of populations of dendritic cells, and I'm not going to go there. Lots of them.